So I want you to picture just a simple ice-cold glass of milk. You probably splash it in your coffee or pour it over your cereal and just assume it's this perfectly standardized raw fuel.
Right. I mean, we treat it like it's completely uniform across the board.
Exactly. But what if I told you there is a hidden, totally invisible, microscopic difference inside that glass? Like a difference that fundamentally dictates how your body actually digests it.
It really is wild. It completely shifts how you view the food you interact with every single day. We just assume our agricultural supply chain is, this monolithic thing.
Yeah, but at the molecular level, there's this tremendous amount of unseen variety, and it carries very real biological consequences.
It really does.
And that unseen variety is exactly what we are unpacking today. Welcome to the deep dive. We're exploring a fascinating scientific study titled Sequence Characterization and Genotyping of Allelic Variants of Beta-casein Gene Establishes Native Cattle of the Dock to be a Natural Resource for A2 Milk.
It's a bit of a mouthful, I know.
Yeah, quite the title. But our mission today is to figure out how, a single genetic letter change in a cow's DNA impacts human health, and why researchers had to travel to the extreme freezing altitudes of the Himalayas just to find a pure source of an ancient milk variant.
I promise you, by the end of this deep dive, you will never look at your morning bowl of cereal the exact same way. We're going to connect the dots between complex cellular biology and, well, the survival of this highly isolated breed of cattle at the very top of the world.
Let's start with the absolute baseline, because before we can talk about Himalayan cows, we need to know what's actually inside a glass of cow's milk. It's not just white water, right?
That's definitely not. It's this massive delivery system. You've got high-quality proteins, carbohydrates, fats, vitamins, minerals. It's incredibly complex.
Right.
But to really get to the core of this study, we have to isolate the proteins. So milk contains about 3.5% protein overall.
Yeah.
And of that total protein, roughly 80% consists of a specific group called caseins.
80%. Wow. Okay.
Yeah, and among those caseins, there's one called beta-casein, which is the second most abundant. It's historically been viewed as one of the most nutritionally important proteins because it just offers this excellent balance of essential amino acids.
Which are basically the fundamental building blocks for human growth, right?
Exactly.
But, for the longest time, I feel like modern nutritional science just viewed those proteins as raw calories. Like, you drink the milk, your stomach acids break down the beta-casein, you absorb the amino acids, and you're done.
That was the old consensus, yeah. But we are completely moving beyond that idea of basic nutrition now.
Because the proteins are actually doing a lot more, right?
Right. We now know that milk proteins have these significant active effects on human biology. As they break down in your digestive tract, they don't just disintegrate into fuel, they release these specific biologically active compounds called bioactive peptides.
Okay, the best way I've heard this visualized, and this is for you listening, is to think of milk proteins as heavily encrypted biological messages.
Oh, I love that analogy.
Yeah, so when you drink milk, your stomach enzymes act as the decryption key. And as the proteins get chopped up, they release these specific bioactive peptides that they suddenly wake up and start directing traffic in your body.
They really do. That active signaling is profound. Once they're released, these peptides can actively modulate your immune system, influence your cardiovascular health, interact with your neurological function.
Wait, neurological function too?
Yeah, and even shift your overall metabolism. Like milk naturally contains bioactive compounds like lactoferrin, which is known for its antimicrobial properties, and osteopontin, which acts as an immune system modulator.
Okay, so if these milk proteins are actively sending these complex messages to our bodies, my immediate question is, what happens when the cow printing those messages has a slightly different genetic code?
That is the million-dollar question, because they aren't all printing the exact same beta-casein.
Right, I'm assuming there's variation.
Tons of it. There are actually 12 known genetic variants of bovine beta-casein. But the dairy industry, and this study in particular, is primarily focused on the two most common variants, which are A1 and A2.
Okay, A1 and A2. And if I go to the grocery store, they look and taste exactly the same, right?
Indistinguishable.
Yeah.
You couldn't tell them apart by looking at them or tasting them. The difference between them is staggeringly small.
Yeah.
Out of a chain of over 200 amino acids that make up this beta-casein protein, They differ by just one single amino acid.
One single link in a chain of over 200.
Yes.
And just to zoom in on how tiny this change is for a second, this whole difference comes down to a single nucleotide mutation in the cow's DNA.
Exactly right.
It's basically a 3-letter genetic sequence that is supposed to read CCT, but at some point in agricultural history, it mutated to CAT. Literally one typo.
Just one typo, and that single typo occurs at position 67 of the beta-casein protein chain. So because of that mutation, in A2 milk, that 67th position holds an amino acid called proline. But in A1 milk, the mutation swaps that out for a different amino acid called histidine.
Proline versus histidine.
Right. And that swap fundamentally alters the structure of the protein, which completely dictates what happens when your digestive enzymes attack it in your gut.
Okay, so let's use the ropes analogy here because I think it perfectly explains the mechanics of what actually happens in your stomach.
Go for it.
So imagine the beta-casein protein as this long, thick rope. And right at position 67, there is a knot tying two pieces of the rope together. In A1 milk, that histidine acts like a really loose slip knot.
A very fragile bond, yes.
Right, so when your stomach enzymes hit that slip knot, it unties almost immediately, and the rope just snaps in two right at that exact spot.
In biochemistry, we call that gastrointestinal proteolytic digestion. The enzymes cleave, or basically sever the protein at that specific histidine site.
And when that rope snaps, it drops this very specific 7-amino acid fragment into your gut. And that fragment is a bioactive peptide called beta-casein 7, or BCM7 for short.
Yes.
But if you are drinking A2 milk, that proline at position 67 acts like a tight, heavily secured square knot, right?
Exactly. Your stomach enzymes just can't untie it. The rope stays intact at that specific spot, preventing the cleavage entirely. So instead of dropping BCM7, it generates a larger, different peptide called BCM9.
Okay, so the structural integrity of that square knot is basically the crux of this entire scientific debate surrounding A1 and A2 milk?
It absolutely is. The A1 slip knot releases BCM7. The A2 square knot does not.
Yeah.
And the reason researchers care so incredibly much about BCM7 is that it is a recognized opioid peptide.
Okay, wait, I want to pause here, though, because it's really easy to hear the phrase opioid peptide and immediately panic. We aren't saying a regular glass of grocery store milk is a dangerous narcotic, right?
No, absolutely not. It is a risk factor being intensely studied, not some classified toxin.
Yeah.
But the biological mechanism is very real.
How so?
Well, because BCM7 is such a small peptide, it has the ability to enter the human bloodstream. And this is the crucial part. It can cross the blood-brain barrier.
Oh, wow.
Yeah, and once it does, it can bind to specific opioid receptors, specifically the mu-opioid receptors, which are located in the central nervous system, but they're also highly concentrated in the human gastrointestinal tract.
So how does binding to those receptors actually affect someone digesting the milk? What does it feel like?
In the gut, opioid receptor activation generally slows down gastric motility. It literally slows down the transit time of food.
Oh, so it just sits there longer.
Exactly. And this is why epidemiological research has explored associations between consuming A1 milk and releasing BCM7 with digestive discomfort. People often experience bloating or altered bowel functions, and they just mistakenly assume, oh, I'm lactose intolerant.
When really, they might actually be reacting to the BCM7 slowing their digestion down.
Precisely.
And researchers are looking at effects that go way beyond just a stomachache too, aren't they?
They are. The scientific literature has been investigating potential epidemiological links between BCM7 and more severe conditions. We're talking cardiovascular disease, type 1 diabetes, and even neurological conditions like autism and schizophrenia.
Wow, from a milk protein.
Yeah, primarily driven by underlying inflammation pathways that the peptide might trigger.
We do have to be incredibly careful here, though, just to keep the science grounded. Epidemiology shows us patterns and associations across large populations, sure, but we don't have definitive clinical cause and effect proof yet for those more severe conditions, do we?
We do not. That's a very important caveat. It is an evolving science. We still need high-quality, randomized clinical research to move this from an associated risk factor to a definitive cause.
Right.
But, the researchers behind this study recognize a very practical reality. If BCM7 is even a potential risk factor, and A2 milk naturally prevents its release entirely.
Then we need to locate and secure natural populations of cattle that only produce the pure A2 variant.
Exactly.
Which brings up a huge logistical problem. If Western grocery store milk is heavily saturated with this A1 slipknot mutation, how do researchers even establish a baseline? Like, where on earth do you go to find a cow that hasn't been touched by modern industrial breeding?
We have to look back at the historical timeline first.
Yeah.
A2 is actually the original ancestral genetic blueprint. Millions of years ago, all cattle produced A2 milk.
So where did A1 come from?
The A1 mutation happened spontaneously in European herds thousands of years ago. And because early farmers were selecting cows based on visible traits like, overall size or how much milk they yielded, they unknowingly began multiplying this invisible mutation.
Because they were just breeding for volume, completely blind to the fact they were passing down this C18 nucleotide typo to millions of future calves.
They had no idea. Industrialized agriculture then took those high-producing breeds like the Holstein Friesian and Jersey cows and just spread them globally.
Right.
Those breeds possess overwhelmingly high proportions of the A1 allele.
Okay, so they had to look outside the industrial system.
Right. To find the unmutated original genetics, the researchers in this study had to travel to one of the most isolated regions on the planet, the Trans-Himalayan region of Ladakh in northern India.
They went to an altitude ranging between 2,300 and 5,000 meters above sea level to study these native Ladakhi cattle. Now, just to clarify for everyone listening, the A2 gene isn't some superpower that helps these cows survive the freezing cold, right?
No, no. The A2 gene itself doesn't provide thermal insulation or anything like that.
Okay, so it's just that their extreme geographic isolation has protected their bloodline from Western agricultural tampering.
That is correct. Their value to this study is entirely based on their isolation.
Yeah.
But the environment they survive in is exactly what kept them isolated. I mean, we're talking about hypobaric hypoxia.
Which is severe lack of oxygen.
Right. Add to that freezing winters, vast barren landscapes with incredibly poor quality vegetation, low humidity, and intense UV radiation.
A modern commercial dairy cow bred for volume would just rapidly perish in those conditions.
Oh, they wouldn't last. Yet the native Ladakhi cattle are practically eating dry scrub brush in freezing temperatures, and they're still producing around 2.5 to 4.5 kilograms of milk daily.
Which is an absolute lifeline for the local human populations there.
It is. It provides critical animal protein during the harshest winter months. So for the scientists, these specific, highly resilient cattle represented a potentially untapped genetic goldmine.
Because they've been physically cut off from modern commercial breeding programs for centuries.
Exactly. The hypothesis was that they might still possess the pure original A2 genetic blueprint in its entirety.
But, I mean, a hypothesis requires proof. You can't just hike up a mountain, look at a cow, and know its genetic makeup.
No, you definitely cannot.
The researchers had to physically travel village to village across the Ladakh region to collect blood samples from 85 native cattle and then bring those samples all the way back to the laboratory to extract the genomic DNA.
And the laboratory methodology here is vital to understanding the results. Once they extracted the DNA, they had to isolate the complete beta-casein gene. They relied on two primary molecular genotyping techniques.
Okay, lay them on me.
DNA sequencing and a process known as PCR-RFLP.
Okay, as someone who doesn't spend their weekends in a genetics lab, you're going to have to walk me through how you actually find one single misspelled letter inside millions of strands of DNA. I know PCR stands for polymerase chain reaction, but how does it actually help researchers spot the mutation?
Think of the animal's DNA as a massive, multi-volume encyclopedia. The single-letter mutation we are looking for is buried deep inside one specific paragraph of one specific page.
Okay.
It is physically too small to observe on its own. So PCR acts as an incredibly precise targeting system. It locates that specific genetic paragraph that codes for the beta-casein protein, isolates it, and then chemically forces it to duplicate itself millions and millions of times.
Oh, I see. So instead of searching the entire library for one tiny typo, you use this biological search function that copies that one specific page a billion times until you have a sample large enough to actually interact with.
That's a perfect way to put it. And once they have that massive amplified sample, they apply the RFLP, which stands for Restriction Fragment Length Polymorphism.
Okay, another mouthful.
Yeah, science loves its acronyms. But this involves introducing a specific restriction enzyme. In this study, they used an enzyme called TAC-I.
Now, I've always heard enzymes described as molecular scissors, but given how specific this test is, that feels like a cliche that doesn't quite capture the mechanics. It sounds more like a highly specific keycard reader at a secure building.
That is a much, much more accurate way to visualize it. The TAC-I enzyme acts as a biological keycard reader that scans the sequence of letters in the DNA. It's programmed to unlock or cut the DNA strand only if it encounters the exact genetic sequence that includes the A1 mutation.
Wow, okay, so if the cow has the pure A2 sequence, like the original blueprint without the typo, the key card just fails, and the enzyme passes right over it without making a cut.
Precisely. The researchers then run the resulting DNA through a gel so they can physically see the fragment sizes.
Okay, so what are they looking for in the gel?
Well, if they see one large uncut fragment measuring exactly 251 base pairs, they know the enzyme couldn't cut it. The cow is pure A2A2.
Got it.
But if the gel reveals smaller, severed fragments of 213 and 38 base pairs, it proves the enzyme found the A1 mutation and cut the strand.
Okay, there's one technical detail in the methodology I wanted to ask about before we get to the results. The study noted that during their sequencing, the, quote, 5' untranslated region of the gene was completely conserved.
Yes.
Wait, untranslated region, doesn't that mean a part of the DNA that doesn't actually code for the milk protein itself? Why would it matter if that part is conserved?
It's a really great observation. Yes, the untranslated region doesn't code for the final amino acids, but it regulates how the gene is actually expressed. Finding that this region is completely conserved, meaning it had zero variations across the samples, indicates this profound genetic stability in the population.
So they haven't been mutating wildly over time.
Right. It proves these cows haven't undergone erratic genetic drift. They are a highly stable, ancient lineage.
Okay, so the lab work is done. They've copied the pages, they've run the enzyme key cards, analyzed the gel fragments. What did the genetic instruction manuals of these 85 high-altitude cattle actually reveal?
The allelic distribution findings were incredibly striking. The Ladakhi cattle demonstrated a massive predominance of the A2 allele, sitting at a frequency of 0.90.
0.90. So 90% of the overall gene pool is the pure ancestral variant.
Yes, and when we break that down by individual animals, 79% of the cattle tested were homozygous A2A2.
Meaning they inherited the pure A2 gene from both parents. They only carry the original genetics.
Exactly. But the most significant data point in the entire study is this. Out of all 85 animals tested, absolutely 0 animals had the A1A1 genotype.
None of them were completely mutated.
Not a single one.
That totally confirms the hypothesis that this isolated population is a massive natural reservoir for A2 milk. But wait, I'm catching a little detail in the math here.
Oh.
Yeah, if 79% were pure A2A2 and 0% were pure A1A1, what about the remaining 21%?
Ah, yeah. That remaining 21% is the crucial plot twist of this research. Those cattle were A1A2. They were heterozygous, meaning they carry one ancestral A2 gene, but they also carry one mutated A1 gene.
But if these cows are completely isolated in the Trans-Himalayan mountains, where did that A1 mutation come from? I mean, it couldn't have just spontaneously appeared at a 21% rate.
It didn't. This reveals a very modern, incredibly real threat to this native population. In recent years, local agriculture has faced intense pressure to increase daily milk production just for basic food security.
Sure, people need to eat.
Right. So to achieve higher yields, some local farmers have begun crossbreeding their hardy native Ladakhi cattle with exotic Jersey cows imported from outside the region.
And as we established earlier, Jersey cows are one of the Western breeds that are heavily saturated with the A1 mutation.
Exactly.
Man, so by trying to get more milk to feed people in the short term, they are accidentally overwriting this pristine ancient instruction manual.
The crossbreeding is slowly introducing the A1 mutation into a genetic pool that has literally been cleaned for centuries. The researchers noted that while an A2 allele frequency of 0.90 is exceptionally high, it's actually slightly lower than the average frequency found in other native Indian cattle breeds, which generally sit around 0.95.
Wow.
Yeah, the data strongly suggests this slight drop is the direct result of the recent influx of Jersey cattle genetics.
It is a profound conflict. I mean, the study proves unequivocally that the native Ladakhi cattle are an invaluable natural resource for A2 milk. They're basically a living repository of an original genetic blueprint thriving in one of the harshest environments on Earth.
They are.
But the study also acts as a blaring alarm bell. Because if this crossbreeding continues unchecked, this incredibly unique genetic adaptation will just be diluted and permanently lost.
The authors of the study make a compelling plea for immediate, systematic conservation efforts. They emphasize the critical need to generate awareness among policy planners, stakeholders, and, the local farmers themselves.
But they can't just tell them to stop breeding for food security entirely, right?
No, they acknowledge that in areas where crossbreeding with Jersey bulls is deemed absolutely essential for immediate food security, there must be strict protocols. They recommend meticulously screening all breeding bulls to ascertain their A1 or A2 genetic status before they are ever introduced to the herds.
Oh, so they just make sure they only use A2 Jersey bulls.
Exactly. This is the only way to minimize the dissemination of the A1 allele into the native germplasm.
It requires such a delicate, highly managed balance. You want the local farmers to have enough milk to survive the winter, but you cannot afford to destroy the ancient genetics that allow these cows to survive at 5,000 meters in the first place. Nor can you lose their status as a pure source of A2 milk.
Right. Understanding our agricultural biodiversity isn't just about preserving history. These native populations hold genetic baselines that we might desperately need as we learn more about human nutrition.
Which just brings us back to the importance of looking closely at the systems we totally take for granted. We assume our food systems are thoroughly mapped out, yet we are still uncovering how minute genetic shifts deeply alter our biological interactions with our food.
We're barely scratching the surface.
Seriously. So to bring it all back home to you listening, the next time you open your fridge and pour a simple glass of milk, remember the invisible universe swirling around inside it. Remember the intricate shapes of microscopic proteins, the difference a single genetic letter can make to your digestive receptors, and the incredibly hardy cattle braving the freezing, oxygen-deprived Himalayan winters to produce it.
It's incredible to think about.
The story of our food is infinitely more complex than the nutritional label on the side of the carton.
It really is. And I'll leave you with this final thought to mull over today. If a single random genetic letter change in a European cow centuries ago somehow altered the neurological and digestive pathways of millions of people drinking milk today, what other tiny, completely unnoticed genetic shifts in our modern agricultural plants and animals are secretly reshaping human health right at this very moment?
Man, that is wild to think about. We'll see you on the next deep dive.